Okay, thanks everyone. Hello and welcome everyone in person and online. Thank you so much for joining us today. This is the first Junior Faculty Business Development Hub workshop that we're hosting. So thank you so much for attending. I'm Britt Hansen. I'm a postdoctoral translational scientist at GOSH, one of the steering committee members for the Business Development Hub. So just to get a housekeeping matter out of the way, if there is a fire alarm and you're in this room, please can you head outside, turn right and right again. So avoid the stairwell and the reception area and head outside, but there shouldn't be one. So hopefully we'll be fine. And then just to let you know that the meeting is being recorded. So hopefully everyone's okay with that. So we're delighted to welcome Zoe Hewitt here today, all the way from Sheffield. So thank you so much for traveling down to be with us today. Very briefly, Zoe is the co-founder and CEO of Regenerative Cell Therapy Consulting, or Regen CTC, where she plays the role of lead quality management specialist. But I will provide more of an introduction later. First, we're going to kick off the session by hearing from Federico, who's a production manager at the GOSH Cell and Gene Therapy Service. So Fred started his career in the production department of an active ingredient manufacturer. Then he moved to manufacturing of monoclonal antibodies for 5 years. So that's IgG and coagulation factors, both upstream and downstream processes. Fred then entered into the advanced therapy medicinal products world 6 years ago, working for 5.5 years in catapults, including at GSK and Achilles Therapeutics, and then joined the GOSH Cell and Gene Therapy Service 5 months ago. So thank you for coming today, Fred. Yeah. Take over.
You might have to use the arrow keys just to...
Okay, that's fine. Good afternoon, everyone. Everyone okay? Yeah? Welcome to the building. Does everyone work here or in the building? Okay, except welcome to the building.
Thank you.
All right, so unfortunately, Barry couldn't be with us today. You will have way more background as to how the facility was developed, but hopefully I will go through a few slides, and if you have any questions, I'll be more than happy to follow up. There's my email at the very end, so if anything is needed, we can discuss at a later time. So we are here to talk about the manufacturing of ATMPs here at GOSH, and if the computer worked, I could change something. So should I just announce it?
Maybe it should work with the arrow.
Click on the screen.
Oh, yeah, if you click on this.
Yeah, there it is.
The arrow should work now.
Okay. So the manufacturing of gene therapies here at GOSH started in the early 2000s, so 2001, with the manufacture of the first SCID patient here at GOSH. The picture on the right, it's actually a lab that you'll be happy to hear that is still live, up and running. We actually have a team in there today, and it has been used for the last 20 years to manufacture all sorts of products. So more recently, because obviously this is a very small lab and it was getting outdated, we needed to build something new, and that's where this fancy new building comes in place. So ZCR was built, and on the very top of the building, so the 4th floor, we have a total of 7 clean rooms. So the clean rooms cover both grading. So we have clean rooms graded as B and clean rooms graded as C. And we also have all the ancillary areas that might and are required to support that manufacture. So that covers waste treatment areas. It covers labs where people can provide a certain degree of training, simulation rooms. We also have storage for our drug substances and drug products upstairs. And the forecasting is that we could manufacture up to, alongside the labs in CTL, we could manufacture up to 150 ATMPs per year, which is an incredible number. Now, to manufacture those 150 products per year at maximum capacity, there's obviously several considerations that we need to take into account. If we think from a contamination control strategy point of view, we need to segregate how those products are being manufactured. There are several ways of doing it, and no solution is a single layer. It's a multitude of different solutions that, when put in practice, will make that happen. But from the diagram, what you're looking at right now is the diagram of the 4th floor. You'll see different colors. And one of the engineering controls that we have in place is that each of these colors actually represents one unique air handling unit. So that allows us to, within the same facility, have completely segregated the air that comes in to each of those clean rooms, and it goes out. And that allows us a multitude of different things. So we can, for example, as we currently do, we can manufacture downstream of AAVs, whilst at the same time having other projects in the vicinity, yet being kind of enveloped and not putting each other at risk, obviously, in addition to other mitigation strategies. Now, what this picture doesn't show you is how it actually looks upstairs, right? That is just a diagram. So we have a couple of pictures to show you what it looks upstairs. It's a series of clean rooms. So as you can imagine, they will be extremely clean and neat and tidy, which is one of my favorite things of working in clean rooms, because isn't it such a beautiful environment to work in? I think it is. But yeah, so on the left-hand side, we have a picture of the corridor that on the previous diagram, it's this corridor in light blue. And on the right, we have a team of our colleagues working in one of the rooms that is highlighted in green. So yeah, you'll see that the room has a series of equipment in there. So I'm not sure if everyone's familiar with this kind of thing, but for example, from the left, we have the isolators. You might have heard the terms before. Isolators, they are transversal to this type of manufacturing ATMP world, and we've been doing it for many, many years, because if you remember the first picture of that lab that has been running for 20 years, the glove ports on the left are actually of an isolator. So isolator has been carrying over with us over the years. We have the prodigies, which I'm sure the majority of you will be familiar, etc. But now we have a clean room, we have equipment in there, but is that all that is needed for us to manufacture? It's not. So there are several things that we need to take into account, and this kind of simple version of a diagram, it's actually covering one of the most fundamental parts of any GMP operation and any operation that wants to be robust and be reproducible. And that is covering the qualification and validation of both the facility, systems, and equipment. So this is a very, it's a complex diagram, so I'm not going to go through every single step, but we have to think from the point of view of having control since the very beginning. So if we are setting up a facility, we want to make sure that the facility is doing what we expect it to do. And the way we control, one of the ways of controlling it is by qualifying and validating all these different areas, the qualifying equipment. And it goes from the very basics. So it can be, because halfway through you'll see dimensions of IOQs, for example. It could go from something very basic, like, does the equipment plug to the plugs that we have in the building? That is something that we might take for granted, but it's something that we need to make sure that it's working as expected. And from that, we evolve all the way to, okay, when are we using this equipment? If we use it across a number of times, are we always getting the same output from the equipment? And all those controls put in place will make sure that the facility, the equipment, and the systems are operating as they expected. And therefore, we know that we have the baseline to then evolve into other considerations, knowing that the baseline and the framework is there. So, yeah. So after doing all of that here at ZCR, which I wasn't part of, a few years ago in 2021, we submitted our license variation. A few months after that, in July 2022, we had NIHR coming around to inspect us. The inspection took place, a few comments went back and forth, and we received the license for ATMP manufacturer in January 2023, which was followed later in the year in October by the license to also manufacture vector. And now that we have a facility and a license, we are then able to do what we all want to do, which is to manufacture and support different companies, different projects at different stages, to actually make these products, put these products out so they can help the patients that we so much want. The way we have set up the operation here allows us to be quite flexible in the way we receive new collaborators and new projects. We have our own in-house quality assurance, in-house QC, in-house manufacturing team, and that puts us in a situation where if someone has a project, they have a manufacturing team, but they might not have the quality aspect. We can absorb those projects by providing them advice and services on the quality aspect of things whilst they manufacture with their own manufacturing teams. Or if you just have a project that you don't have the manufacturing team, the quality, we can absorb those projects as well just by providing all those different types of services. That is currently ongoing with a few projects. Here I have a couple of the UCL projects. So we have the CD34 team that has a platform approach for all those conditions. And we also have the CARPAL team currently with us. Outside the UCL sphere, we partnered with a couple of commercial companies. So we have on site both Virocell, which are currently manufacturing vector for phase 1 trials. And we also have Lucid that will be looking at manufacturing products for phase 1 trials with CAR-T cells. And I think that is all I had for today, unless you have questions or are we gonna share the slides?
Yeah, we can share the slides for sure if you're happy with that.
Yeah.
But since you won't be staying on, if anyone has questions, now would be a good time. Yeah.
Thank you. That was really interesting. Could I ask just about that number of 150 per year?
Yeah.
Which is obviously based on some timeline for each product.
Mm-hmm.
So could I sort of ask about that? What's the timeline for most of the ones that you have going so far?
Yes,
So I mean, because I'm obviously some cell products would have a very long timeline. So I'm just interested in what your 150 is based on.
The 150 is
an example.
No, it's a great question, a question that I asked this morning, because the 150 from experience, it can be a number that I'm not sure would be attainable, but that is what the forecast says. I'm not privy to the details of the forecast, so I don't know, but definitely because these therapies and this, for example, the expansion, if you expand for 2 days or 14 days, it will have a massive impact on the amount of output that you can put through the facility. So I'm not quite sure where the figure came from, but I'm more than happy to just try and dig a bit more about that.
And say an existing product, would it, one of the ones you're doing now, would it be taking a couple of months or a month? Or,
if we're talking strictly about the manufacturing, we have products that are way faster.
Way faster. So
that.
It's a very short use of
very
one
short,
of those dedicated spaces.
Yeah, very short manufacture process that we can talk about 3-4 days. And then if you put together the number of rooms that we have, that might be how it was extrapolated to the 150. But yeah, it's definitely dependent on the type of products you have. Of course.
And are you the best person to email if we've got a project that we want to approach? Is it best you and Barry or you or is there an online registration portal or do we just contact you guys?
The onboarding of new projects will have to go through Barry and he will point you towards the right people to speak about that. I support the production, so it's a few steps down the line after the onboarding of new projects. But yeah, but feel free to send questions.
I know that you won't be able to answer this because everybody's really different, but do you have an idea of like average cost per week for the facility, if you wanted to use the facility for 1 week, the clean rooms, how much would that, to give people an idea?
I'm not sure I can share that sort of information. It will obviously be dependent on the project, etc. I am privy to the figures, but I'm not sure if I could share it in this forum because obviously there is contractual agreements with all these projects, etc. But I can definitely ask the question if we wanted to put out there a figure that could work as a baseline for people to take it from there. I can definitely ask that question, but can't give you a figure. So
No, I think that would actually be useful to have that figure, but for people who are then grant writing or kind of even just thinking about a grant, so they have a ballpark figure that they could work with, but it's good to know you can haggle.
okay, I'll pass that information to the person that haggles. Yeah.
Can I just ask you, so all the projects you presented were kind of lengthy projects. AAV capabilities and
We do. So the
titers you need for kind of,
do you mind?
yeah, I'm just wondering about AAV capability to, and you know, generating high enough titers that you'd need for kind of clinical grade.
We do have the room, so each of these rooms can be molded to what you need. So at the moment, they have a set of equipment in there, but if a project was to come forward where they needed some of the space, we could, so the clean rooms, they are not big per se, but there is always a scope to readjust the rooms in a way that they can accommodate enough equipment to manufacture AAV in big, not big, but medium volumes if needed.
Do you have a license for tissue engineering?
Sorry?
Do you have a license for tissue engineering? Like for building grafts?
No, I don't think we do at the moment.
It's something that you could obtain if needed.
That would have to go through the business development side of things, but I don't think there's anything that we can't try. Right? We have the facility, we have the systems in place. If there is an opportunity to think about something else, why not? We can explore it.
I think most GMP facilities, once you have a product licensed in a GMP facility, the variation to your license to add a different project is much easier than getting the license to start with.
Okay.
So a lot of it depends on your process and your product that you're taking to that GMP facility. So.
Thanks for transporting us into the musical. Okay, so now I'm going to properly introduce Zoe. Zoe obtained her PhD in 2001 from the Rosalind Institute at the University of Edinburgh, where she stayed on to develop differentiation protocols for hepatocytes. She then moved to the University of Sheffield in 2006, establishing and managing a clean room facility that was responsible for delivering the UK's first clinically available human pluripotent stem cell starting materials called MasterChef cell lines. Maybe we'll hear more about the inspiration of these later on. And through this work, Zoe and her team worked with pharma and biotech companies to deliver starting master cell banks for the London project to cure blindness, resulting in the first successful UK clinical trial of a cell replacement therapy, which was for macular degeneration. And then in 2015, Zoe took the leap out of research and into project management for the UK Regenerative Medicine platform, working towards advancing regenerative medicine therapies by focusing on the rate-limiting steps. And recently, the second UK hPSC-derived cell therapy entered clinical trial, this time for the treatment of Parkinson's disease. Zoe has clearly got a wealth of knowledge and expertise regarding how to carry academic research through to translational development, and in her own words, by demystifying the process and providing solution-based advice to help ATMP developers to implement a strategic vision that will ultimately enable them to bridge the valley of death and secure translational or commercial funding for these amazing therapies. So we look forward to hearing more about this in your talk, Zoe. We will have a Q&A at the end, but we want this to be interactive, so please feel free to jump in, challenge, prod, ask questions as much as you want, and yeah, thank you.
Thank you very much, Britt. That was a very comprehensive introduction. You've certainly done your background research on me. Some of those things I'd forgotten. So thank you for the invite to come down to London to talk to you today. I am really passionate about demystifying and making people aware around the power of quality management. It upsets me greatly that people think it is onerous and something you can flick down the road and do later when it becomes a huge mountain instead of a little blip. So hopefully by the end of the day, you will all go back feeling empowered to be able to implement some of these strategies really early, which will make Fred's job much easier when you go to him to want to use this clean room. So, spacebar, did you say?
On the slide again.
On this screen or this?
Yeah.
Left, on the left.
Yeah.
Left.
Okay, so I can probably run through this because Britt's done it all for me. So I wanted to give you a bit of background to explain why I'm stood here in front of you today, because I have spent my career inside academia. And so a lot of people look at that and go, well, what will she know about translating a medicinal product? So as Britt said, I started a passion for cell therapy actually back in 1999, which feels like archaic these days, when Jamie Thompson published his paper on human ES cells. I was captivated and I wanted to get them into patients, and that was my driving force behind everything that I've done since. My PhD up at Roslin in 2001 was starting to look already about the challenge of, okay, well, if we've got a cell therapy, how do we make sure it's safe? And I sort of very quickly realised that my passion was about making something safe to go into a patient rather than really understanding the biology. So after I'd done my PhD and I'd worked with Dave Hay about hepatocyte differentiation, my plan was to take this passion and go abroad and work in Australia on thymic epithelial generation from pluripotent stem cells. The idea being that you could make a chimeric human, and therefore when you gave them a cell therapy derived from the same thing, they wouldn't have an immune rejection to it. And this got me through my PhD because I loved the science bench work, I hated writing, but I had this carrot to go to Australia. And I took a job at Scottish Water, where very glamorously implemented their ISO 9001 certification. And then I got the unfortunate news that the grant had fallen through and I wouldn't be going to Australia. So I ended up in Sheffield, not abroad. And Professor Harry Moore said, we built a clean room, make it work. And I said, you built what? And so I spent my career really trying to understand how you build a clean room, what it takes to make it work, and then making that work. I say it was my first postdoc, but it was never a postdoc. I kind of skipped postdocing and went to quality operational middle management and have built my career from there, really. So we did make it work. We have made the master chef cell lines. This was a play on words because we already had the research grade chef lines. The master cell banks that you produce in GMP was where the master chef distinction came from. It just so happens to play on our favourite TV show. So we did make it work. We made 14 clinical grade lines. We re-derived 3 in that environment. We made the master cell bank for Peak Coffee's London Cure for Blindness project, which went into clinical trial. There are now people in London who have my cells in the back of their eyes, okay, converted into RPE cells eventually, but my cells, my lab, my team, my quality management system. And then we were a little bit ahead of our curve in Sheffield, and we created this clean room back in 2006. It actually closed in 2013 because there wasn't enough funding to continue it. It was at a time where really kind of cell therapy hadn't kicked off yet, the university hadn't invested in it, and so sadly most of my team were made redundant. And then I became part of the UK Regenerative Medicine platform, and I originally was their quality advisor, and then went out and had my daughter, and when I came back seemed to know more about what was going on than the people who hadn't left, and so they asked me to project manage it for them. So I then moved into project management. And during the course of that, those 2 UKRP hubs were focused on the translational bottlenecks around taking pluripotent derived cell therapies into the clinic. And in 2022, I established Regen CTC because it was horrifying to realise that I was involved in the first pluripotent stem cell derived clinical trial. And then in 2023, which was when the next one was in the UK, I was involved in that too. And it's like, there were a lot of people working in this space. Why aren't we getting more of them into patients? And that's where the drive comes from, really, for doing that. There's a man waving at me with a piece of paper out in the corridor. Pointing at a piece of paper. Was that the coffee? What do we do with that? Okay, so I hope what my purpose of telling you all of that was really to sort of set my reputation, because I had done all of this in academia, and that's why I'm here today. So policy management then, it's a dry subject, so I'm going to try and make this engaging for you. Shock horror, it's not new. So the concept of quality management isn't something that's even this century. you can trace it back to medieval craftsmen in the 13th century when they built little unions to make guilds where they would then set strict guidelines to make products, and then an inspection committee would come and give them a little stamp to say this is a flawless product or service. And so, you know, as a concept, it should be something that we've got a handle on by now. And that method of simply marking a product as flawless, and then craftsmen started to mark them to say that one's mine, so that you could trace defective. Originally, it was the idea was you could trace a defective product, but actually it became a mark of good craftsmanship. That stood the test of time until the 19th century when the Industrial Revolution started. And you know, quality management systems have fortuitously evolved over that time. Initially, in the Industrial Revolution, it was about increasing productivity, unsurprisingly, and so quality took a little bit of a nosedive. That focused on having the right people and then inspecting to make sure that they had done a good job and retaining any defects. In the World Wars, safety became obviously, you know, you couldn't send out machine guns or whatever you were manufacturing that were unsafe to use, and so they started to inspect every single one. That became too onerous, so they started to look at statistical sampling, audit, those kinds of things. And actually it was the Japanese engineers in the mid-20th century that started to think about company-wide QC, they called it at the time, which elevated their ability to produce high quality goods at low costs more efficiently than the Americans. And then that concept of whole company QC was basically the foundation of the modern day quality management system. So it's been around a while. And just to put that in context, ISO 9001, does anyone know when that was established? It's younger than me, it was 1987. So it was 1987 when we suddenly had a harmonized way of doing quality management across different sectors and things. And that's been, you know, that's been modified a few times even since then. So, you know, we can do this because it's been done for a long time. Well, why is quality management important? Everyone knows it's important, you know, and yet we put it off. And I guess really for me, you know, thalidomide is the one that everyone thinks of. So, you know, a drug developed in the 1950s that was initially used as a sedative, tranquilizer, and then everyone claimed it was safe for people who were to treat morning sickness. And we know where that led us to. And that led us to the 1968 Medicines Act, which is the foundation for what we're all trying to do. It resulted in the Orange Guide, its first iteration in the 1971, which we're now on like the 11th edition. And I guess the reason for saying this is it is these awful incidents that cause legislation to change. And I don't think I have to preach about this to this audience, but you know, we're still shaping that. There are still things that go wrong. Making new medicines is not without risk, we know that, but some of these things, this is why we control stuff. It's why it's important. And you can have these slides afterwards, so I'm not going to go through these slides, but you know. So to summarize then this little bit, quality management principles are not new. They have evolved over time. They will continue to evolve over time as we learn from our mistakes. And that is really the purpose and the hope for a continuous improvement, which is what every quality management system should be set up to do. It's record, improve, adapt, change, implement. So for me, there are 4 simple principles at the heart of quality management that I think if everyone can think about these in everyday life, or in their work anyway, then we'll be in a better place than we are now. And they're simple and obvious, really. You control what you can control. You have traceability, have transparency, and take responsibility. And if everything you do has those 4 principles at the heart of them, then I don't think you'll be doing something too far wrong. If you can adopt those early in a program of product development, you will benefit greatly as you move through to Frederick's GMP facility. And he will very much appreciate it when you get there, if you've got some of this stuff in place. Okay, so this is my statement that I made. We need to do quality management because we've got to submit a regulatory dossier. That's factually correct. You will need a dossier and you will be expected to have considered quality management. But that's not the time to consider it. If you're only considering quality management when you're about to submit for regulatory, then you're in big trouble. Or even if you're starting to think about it when you're going for these really big translational awards, because they want to see that you've already started to think about it. Some of these things can be implemented much earlier. You know, in that earlier implementation, it provides you with power. You know, it provides you with evidence that supports your experiments, that supports the traceability of why you've chosen to do things in specific ways. It allows you to really pick when something goes wrong, it allows you to investigate more thoroughly what could have gone wrong and prevents us from just repeating something over and over again, going, why isn't this working anymore? You can really start to unpack that. Ultimately, that saves time and resources. And more importantly, when you start to look at investment or spinning out a company, it allows you to show you're in control and it adds value to what you're trying to get investment into as you have confidence in what you're trying to sell, essentially, ultimately, whether that to be to a regulator or to an investor. I was asked to talk a little bit about development pipelines. This is a horrible slide, sorry. It's a very nice slide, it's one that I've taken from somewhere. And it's really challenging to do this because ATMPs are such a diverse group of products. And so as we were just discussing, you know, what takes a couple of weeks for one product can take several months for a different product. And therefore the development is different. So they are much more complex than small molecule therapeutics. They are diverse. The average time to develop one takes about 15 years, I am told. Some of them longer, some of them shorter. There is a lot of resources out there, and this is actually taken, again, you can have these slides, and there's some links through here to the ATTC toolkit, which is NHS readiness type. How do we roll out these therapies within the NHS? The ATM roadmap, this is taken from, goes through all of these different sections and points you to the groups of people that you should talk to and when. And I think my advice is to take advice early, take lots of advice. Even within the regulator, there's variation in what you're told. You absolutely need to know your product and know what advice you're asking for when you go to speak to them, because they will not tell you how to do it. They will tell you whether what you're proposing is acceptable or not. And they might give you advice on, well, have you considered this? But they will not tell you how. Knowing your products, there's many tools like this. Again, this is for you to have a look at later, but you know, just it's a flow diagram to work out what product you are making and what it would be classified so that you can make sure you get the right kind of advice when you go. So again, I'm not going to dwell on that, it's for your later use. So rewinding, I'm here to try and educate early innovators around quality management, what they should think about. So it's very daunting to think about the end, but you have to think about the end when you're at the beginning, because ultimately that's where you're trying to get to. And I think one of the take-home messages that I would like the room to take is, you know, there are decisions that you make very early in the lifespan of an ATMP that can have very dramatic consequences for whether it can be used later. And so having the end in mind is really important. You will not have all of the answers to all of the questions, or all of the documents, or all of the evidence early on. But if you're starting to think about it, you will just naturally start to collate more information that will end up being useful. But it takes time. So when you're thinking about implementing a quality management system, I would say that there are 4 elements to start with. This would be where I would start. So the images for change management, sorry, I couldn't find one that was more suitable, but it amused me a bit. So good documentation practice is number 1. It is fundamental to everything we do. It is irrelevant whether you're taking something to the clinic or you're just doing a publication. Good documentation practice is the foundation. And I wholeheartedly believe it's something we are failing at teaching at undergraduate level, postgraduate level, and therefore it comes as a big shock. Risk management, but specifically quality risk management, is the second element I would put in place, because that determines how you then assess event reporting, or is it the impact of an event or the impact of a change. So you have to be able to assess the impact of what you're doing, and so you have to define how you're going about doing that. And that should be with the product in mind. What are you going to use it for? How are you going to use it? And how is it going to be effective? So I was going to take each one of these a bit at a time, one at a time. So document practice, I hate lab books. They are. So for me, a lab book is not an effective quality management tool. And I think a lot of that is because we don't teach people how to use them properly, is my biggest bugbear. But also they're almost like a diary. They're a chronological series of events. It doesn't, for me, doing cell culture, it's much better to think about my cells over time, rather than comparing one cell culture with another cell culture with another cell culture on the same day. So I used to work with product files or folders, and I kept projects separately. They can be effective to support due diligence, but we have to standardize and give training on what we want to put in them. And without that, they are just a to-do list. And as somebody who has spent way too many hours trying to find nuggets of information, they basically aren't worth the paper they're written on. So having said that, training doesn't have to be onerous, but it should be consistent. You should give everyone the same training, and you should review to make sure that the training has been effective and people are actually following it. So this comes to the PI now. These are things that you will have to put in place in your group. Now, from my perspective, I promised I would give solutions. So there's a couple of things that I think you could consider doing. They're very simple. You give a checklist. So decide what information you want, write it down on a piece of paper. When you get a new PhD student, you hand them the lab book, you hand them the piece of paper, you say, this is what I want you to record. And then you make sure within their first few weeks or months that you're checking that that is what they're doing. Then they go, oh, I need to write this down because it's going to get checked. And they change the way they think about it. The alternative, if you don't want to use lab books, is to provide templates. So we used to have a passaging record. So every time somebody passaged a set of cells, it had exactly it had the label that you had to write in. It had exactly which media you'd fed it, you were going to use, the passaging reagent, the matrix that you were going to use, the new label you were going to give. Then on the back of that, it had the observations for the next week. And then it linked to the next one, and you copied them over. And then they were all PDF'd and scanned in. We had a hard copy that in the lab you could look at, and we had an electronic version in case anyone. So you could do that with any assay. There are certain things you fill in all the time. It's easier to fill in a box where you know what you've got to write than it is to have a blank page and go, right, what do I need to write on this blank page? So there we go, solutions. Alcoa. How many people know what Alcoa stands for? Heard of it? It's one of the most fundamental things. So Alcoa, and then it becomes Alcoa Plus as you start to go into GMP. So, this is something, if you can implement this early, set you up for the development. So when you write data or an entry or a record, it needs to be attributable to somebody who's written it down, which means you have to either initial it or you sign it or you put your name against it. So somebody knows that data belongs to you. That's the first one. It needs to be legible. So lab books not always legible, handwriting not always legible, electronic notebooks are more legible. But it needs to be written in a form that isn't going to fade over time. So think about the type of pen that you're using. Don't use pencil for obvious reasons. Contemporaneous. How many people in this room have written on a piece of blue roll or a glove and then copied it up in the lab book later? Yeah, everybody. It's not contemporaneous. It's not written at the time. It's not time-stamped. It's actually falsification of data, because there is nothing you can prove that when you copied it up correctly if you then throw the glove away. What you should in theory do is stick the glove in the lab book, sign it, or copy it up and just get your pal to go, that the same? Yeah, sign it, say that you've copied it up, or just write it in the lab book to start with and not on your hand. So yeah, contemporaneous. I'm very proud that I can say that word because I couldn't for a long time. Original needs to be you need to describe the data or the experiments, exactly how it was done, and retain that. So if you then go on and do data analysis on some results, you should make sure that you have the original untouched version somewhere that someone else can go back to. If you're working in a lab book and these things are electronic, cross-reference where they're filed, please, because written in a lab book to say, yeah, it's filed electronically. Where do I go and look for that? So help the next generation out, because to be honest, it will help yourself out, because when you come to write up your thesis at the end, you can't remember what you did on day one. And so actually some of these things, they'll just help you with your PhD if or your publication, as well as if you choose to take some of this stuff into the clinic. And then the last one is accurate, which goes back to copying up the information. So it needs to be error-free and it needs to be truthful. And if you have made an error, a single line through it so you can still read what was there, an initial to say that it was you that's changed it, and then a justification for why you have changed it. And then a date, please, just to say. Yes.
Yeah, this all makes a lot of sense. I have a couple of questions. So in terms of the lab book, if it's electronic, I think that's more challenging because a lot of labs don't allow kind of electronics in the lab. So then that isn't going to ever be contemporaneous. You're kind of going back. So would you recommend then just sticking to?
So if you're going to have an electronic lab book, there should be a dedicated place within the lab where you can access that.
Okay.
So
yeah.
you could have a computer in the lab that never leaves the lab, that you can use with gloves on, that you can use in real time. I think some of the problems with electronic lab books are they're off-the-shelf systems, so they feel a little bit constrained. I've never used an electronic lab book. I was actually told when I was implementing our quality management system that because it didn't support the students, that the IT department wouldn't support me to implement it. So we just did everything on paper and then scanned it in. It's a perfectly reasonable way of working in my book.
Yeah, it's in my experience, it's challenging then for someone coming after you to then find information if it's all been written and then scanned. So yeah, how would you go about?
So that comes down to so we will get to that. It comes down to good metadata in your filing structures.
Okay.
So I think my basic rule for this part is if it isn't written down, it didn't happen.
What about audio or video recording?
Sorry?
What about audio or video recording?
Video recording.
Video is fancy, but audio.
Audio.
Yeah. I'm doing this now and
yes, I guess that comes down to filing it, and then if you have to, it's also about, there's no point making documents and records if you can't retrieve the information. So if somebody's going to sit through 4 hours of you talking into a lab book, are they ever going to do it?
be reporting back on written something,
Yeah.
so we come back to the
Okay,
contemporaneous.
so if it's recorded in real time and it's recorded and then it's written up to say exactly where it is and then what you covered in your audio record so that somebody can look for it. I think for me it's about retrieval of the information, because if it's in a system that you can't retrieve the information, you might as well not write it down.
There is technology to do that.
Yes, and you know.
Now we can.
Yeah, yes, I know. As my daughter keeps telling me, I'm archaic. So yes, no, it is. But you have to think about as well, it also needs to be secure. That's the other thing. So can anyone else have imitated it, sliced the video or the audio, stuck it to like all that kind of stuff? That will come in as you get later. But for R&D, you don't necessarily need to be that fancy, I guess. But if it's easier.
Can I just ask about the attributable? Is that just so you know who's done what, rather than, because there's obviously quite a lot of turnover in lab groups. You've got an MSc student doing this, a PhD student doing this. It's not that they're still contactable, and do you see what I mean? They could have left 2 years ago.
Invariably people have left, but I think it's so that you can connect who's done what, and you can look at things like, well, were they trained to do that? Who has made that record, if you need to find more information about it. So if you know it was me, then you might check my lab books, you might check my files, you sort of narrowing down your scope in discovery. But also when you move on through this, if there is a problem, let's say, it might always be the same person. So for example, and this isn't about blame, it's about trying to have the best system. We had a technician who worked in our facility, and every time she wrote down which media had been used, it was like, that doesn't make any sense, because she got dyscalculia and she kept flipping numbers. And we only knew because we knew it was the same person every time. So we were able to then put a mitigation in place that whenever she wrote a record, someone countersigned to just check her numbers. And she was fine with that, but it was just like, okay, we think we need to put something extra in place here. Same, we stopped her from making media because she couldn't tell the difference between one bottle and the other. There were challenges, but you can put things in place to support them, or add additional training, or negotiate that somebody moves into a different position if, like, if a PI kept gardening at weekends and causing fungal infections. So, you know, it helps to know who it is who's doing something. Anymore,
It's more of a general comment, because I think coming back to the digital lab book, it's kind of a big push now at most universities to move to digital lab books. I think just, I guess, as a more general group, to be kind of on top of why that might be. I don't think from the university standpoint, it's coming from the Salcoa standpoint, it's coming from a actual databasing standpoint. So actually there might need to be some training put in place. So like you say, everyone's actually getting standardized training so that the information that's going into that as a translational grouping is actually useful.
And it's also the case that the provision of laptops and tablets in the lab is not provided by the university, who wants electronic lab books to be used, and that is difficult because one in the lab is probably not enough. You know, everybody trying to use one device, and then they're carried around, and then you have to swap them, as well as taking your lab coats on and off. Do an exit a room.
It's not, I don't see what's wrong with a paper and then scan it in, if I'm truly honest. But what I would recommend is, for goodness sake, if you are on a paper-based system, start scanning them in as soon as you make your first record, because it was several months into our paper-based system, actually, and it was when we were audited by a pharma company and they came in and they wanted to procure part of what we'd done. They brought a team in with industrial size scanners and started to scan. I'm thinking, thank goodness I haven't got but I can keep this going. This is a great idea. But once you start the system to digitize it, once you've gone manual, or like do it at the same time, PDFs then are readable, and you can look at them for years to come, and the ink doesn't fade. So they're much smaller to save as well on extra. So this gets back to the point before about retrieval, storage and retrieval. These products are complex, the development pipeline is long, they come with a lot of data. Academia is known for short-term contracts, lots of different people, lots of different systems for filing your stuff. Everyone will have a slightly different brain that works where they recover things from. So as a PI, I think it's important that you provide training. You almost need to say, well, this is the way we file stuff in my lab. Again, it goes back to that lab book, checklist, information that's provided on day one. You know, and you can then retain the knowledge, because at least there's somebody there who can always find the information or show other people. So, and tell everybody. If you can find other people's data easily, you are more likely to look at it and then continue a project. If it's complicated and you've got to listen to 10 hours worth of audio before you know what it is you're supposed to do, you will just go, do you know what, I'll just make it up as I go along, and I'll start from the beginning and I'll do it my way. So then you're getting a lot of repetition. So think about how you're going to trend and easily retrieve data. So again, I promise solutions. As soon as you can, start to get into the habit of making standard operating procedures. So it might be one researcher, one project, and it's your protocol, but write it down. Write it down and then test it on a colleague. If I give you this, do you understand what I'm asking you to? Can you repeat this, please? Because when we write stuff down, there's a level of assumption that you have, because you know what you're doing, that makes it not unambiguous. and it makes it unclear. So if you test it on a colleague, then you know you're in the right ballpark. And then keep it up to date. Use project files or folders with a predetermined filing structure. So, you know, if you're using electronic files, use, Ben, you're laughing, that's off-putting, use a reverse date.
What to be when I try and make project names?
Because the names are ridiculous.
But let's just Pathfinder was a great name for a project. Like.
So yeah, use reverse dates at the start of your file name. That means it will always be filed chronologically. People go, oh yeah, but you can do that on the date it was modified or the date it was. Actually, if it's in the file name, it's easier to use the search function. Put good metadata in your file name. So, you know, have a standard descriptor, so passage record, then have the cell line name, the number of the passage record. So you define all of this in your standard operating procedures. This is how we file data. Have a blank template for a new project. We have this filing structure, project setup, project reagents, project whatever it is for your project. And then have that people copy, paste, and then put all of the data in a way. It makes it easier. And the last one, which I think I've already said, is make sure if you're using a paper-based system, but you've got some data electronically, vice versa, that you cross-reference where that is and where it links to, because microscope images are the bane of my life. So, you know, you take hundreds of microscope images when you're doing cell culture. Trying to relate anyone to an observation in a lab book is an absolute nightmare, because no one knows, well, was that this week or last week? It's the same, because it's the same microscope, and let's face it, cell lines don't look massively different. You might remember in a month, maybe if you've got a good memory, 6 months by the end of your PhD, can you remember which one was which? So we used to mark on our records a photograph has been taken. Just even that, this passage, this observation, there is a photograph, and then because it was filed by date order, by cell line name, we could then go and look at that photograph and you knew which photograph was what. Okay, so that's all I'm going to say. Any other questions or comments on good documentation practice? Which is a few things. There are many others. I could be here all day just about good documentation practice. It's a bit of a soapbox of mine. I'll move briefly on to risk management. So risk management, again, is not a concept you are not all familiar with, but it usually comes from a health and safety perspective, most risk management. Not many people think about what the product that they're developing and the quality risks associated with what you're doing, in my experience. So for this, it's quality risks associated with the clinical use of the advanced therapy, and that means in terms of their safety and their potency or efficacy. You know, what am I doing here, and could it impact the patient receiving this therapy? So, you know, we talked, Fred talked briefly about contamination containment. If I walk around with no shoes on because it's hot and I'm in shorts, I've got all that skin, am I encouraging beasties into my cell culture environment, which means I'm actually increasing the risk of contamination of my cell product, just off the top of my head. There you go. Quality risk management needs to be data-driven, it should be evidence-based, and it should reflect risk-benefit. Consider when you're doing risk management, how a risk is assessed, how you'll assess it, how you'll detect it, how you'll control it, and then how you will reduce it. So you ban open-toe shoes, and you have to have your legs covered when you're in cell culture, and a lab coat on, and gloves, and all those sorts of things. The reason I put this as my second founding thing is because it impacts how you assess impact. So how you think about quality risks will determine how you think about whether somebody has deviated from a protocol and what the impact of that has been, or if you change a protocol, how that will impact the quality and the safety of the product, and so therefore how you assess that. And you know, it's really important when you do any kind of risk assessments that these are regularly reviewed, and reviewed by multiple people ideally, because different people have got different experiences which will bring a different lens to your risk assessment. Seen this before, same kind of risk assessment. This one we've weighted it a little bit so that we think something that is improbable but very serious is more of a risk than something that has no impact but happens more frequently. And I guess that could be product related as well. Again, these are on here not for you to read now, but really just for you to take away. So this was just a couple of things, a way of describing a quality risk management, how you would, the types of consequences you need to consider when you're considering a quality risk. And then you just multiply them together to get the table before. And the idea is that anything you have that is an unacceptable risk, so amber, or an intolerable risk, intolerable, critical, you can name them as you like, just define it in the protocol is what I would say. So you can look at those at your leisure. Okay, more solutions. So people may have heard of a TPP, target product profile, where you define what your product is, pharmacokinetics, that kind of thing. There is also a separate document called a quality TPP, which then looks at the product from a quality perspective. So this is based on your understanding of the mechanism of action, the intended clinical indication, route of administration, and basically you need to think about what characteristics you can measure of your product that would enable you to think about the efficacy or the safety of the product in the patient. So these will be things like, its appearance, for example. Should it be cloudy and look contaminated by bacteria, or should it be clear? What indicators have you got that could impact the potency of your product, and can you define those or write those down? Write as many down as you can, and then as you develop, you hone those and you determine which ones are the critical quality attributes. So which of these characteristics that we measure actually is most closely linked to the success of our product? Or equally, which one can we measure that always indicates a failure? Because then if you can stop early, you save money if you're doing manufacturing. So those things that indicate failure are as important as the things that indicate success. Then consider, the environment you're in, the way we all have things when we're doing scientific experiments that we know when an experiment isn't working. Oh, this isn't going to work, the cells are overconfluent. These are kind of quality attributes that you've already started to consider, you've just not thought of them in that way. So process parameters, if you write down when things go wrong, you can also link those to, does it always fail on a really hot, sunny, humid day? Or is it because in the winter, the university turns the heating off at 6 o'clock at night and I'm still working, and then actually everything fails if I do this really late? Like, can you relate anything to the environment around you? When we get into Frederick's GMP facility, that will all be controlled. But it's important that he knows actually that if there's an excursion in temperature in his facility, that impacts your product. So if you already know that, it's information. And then how you will reduce these. And like I said at the start, I can see a lot of people around the room are going, this is so big, I'm never going to be able to do all of this. You won't have all of these things in place now. But if you're thinking about them now, when you're doing your experiments, you go, actually, no, I'm just going to write that down because that might be important later. Yeah, so that's the purpose of today. It's like, you're not going to be able to put all of this stuff in place now. One of my pleas would be event reporting though. So it's all well and good having defined protocols, but you need to make sure that they are A, robust, and B, being used. And the way we did this is we looked at a reporting system where we just said, look, if you've done anything and it's different than what was written down or what was expected, just write it down. It's not a problem. It was a really simple sheet that we put in every lab that said, what did you do? So I was carrying a stack of 10 flasks to the incubator and then tripped and they all fell on the floor. The next day, one of those is contaminated. That's why it's got nothing to do with the media. I don't have to throw it all away. Or the one lad who I went out and got drunk this weekend and I forgot to come in and beat the cells. That's why they're all being yellow. It's amazing what people will tell you when you don't call it a deviation if you just call it an event. because I don't think it's important, but you know, trending those is really key. It helps support investigations, it helps to support process improvement and safety of your product, but it does require strong leadership and a positive culture to learn, not to blame. So again, it's about encouraging staff to report the fact that they didn't come in at the weekend and feed the cells because they went out and got drunk. That was probably not ideal. It was his first week, but you know, he felt comfortable to come and tell me that. So it was like, right, okay, well, we know why they all look very yellow. We don't have to decontaminate the lab. And if you've got a positive culture and you've got strong leadership, you can empower staff and you will get staff engagement, and then they will just write it down because they know it's not about them, it's about improving the processes for everybody. So this is how we defined an event, any action or occurrence which was different to what was expected or written, irrespective of impact. And the irrespective of impact, it was to really encourage people to report near misses, because near misses are actually really excellent learning opportunities. And they also help not just continuous improvement, but also to validate whether your monitoring and control strategies are actually any good. Because if you get a lot of near misses and no events, then you're doing something right, actually, because it's showing that you're witnessing steps or you're double checking, countersigning, whatever it is that you put in place is actually catching a lot of these things before they become a problem. And that actually near misses then just encourage more training. Events, once something has gone wrong or it's been done differently, then you need more control strategies to stop that from happening. Yeah, and a lot of events people seem to think they're out of their control. It's not my fault the delivery driver keeps sending it to this place down the street. Well, have we had a conversation with the delivery driver? Have we contacted the company to say, excuse me, the last 5 times you've delivered this really sensitive piece of material to me, it's sat somewhere else for 2 weeks before I've got hold of it, that's not acceptable. Maybe don't use that supplier anymore. If it's your only supplier, then you have to put things in their contract about the supply of those materials. So there are always actions even when you don't think it's your fault. I'm not saying you'll always be able to solve them. I'm not saying it's always going to be easy. Make event reporting simple to do. So like I say, this in situ log in the lab where people write it down, but then going back to those 4 key principles, there has to be ownership, there has to be responsibility for picking those up and reviewing them. And then looking at, okay, there's one on this one, and there's one on that one, and that one. They're all minor, but they're all the same. There is a bigger problem. So somebody has to collate that information. You have to make that part of somebody's role to do that. You can't just expect someone to do it, because unless somebody is responsible for doing that, it won't get done. And then you might as well not bother recording them if no one's checking. Change management. Change management, sometimes called change control. It's the most hated, these first 4, I'll be honest with you. It's the one that everyone is frightened of because they think it's onerous to do and it doesn't apply to them because they're doing research. But it's key. And actually, if it's done in a stage appropriate way, it's very doable. Especially small teams think it's onerous because there's not very many of them. Usually change control in pharma at the end, you know, you will want to make a change. It has to be reviewed by quality, have to be signed off by somebody else, make sure that it's not going to impact the product. That's right, because at the end, when you're making a product, you don't want people to change it willy-nilly because it's got all of this supporting data. So that by the time you're producing a medicinal product, change control is fundamental to making sure you can reproducibly and robustly provide patients with the medicine that they deserve. In an R&D setting, you're not there, but it still has its place, I would argue. So yeah, I do research, I can't control things. Science is kind of based on controlling things, I would argue. Control variables all the time. So what's, you know. So if we can get to a system where you think about how to capture those changes that you've made to your protocol. So you go back to your protocol that I've said, right, there's one, in fact, it's the next slide, I believe. No, it isn't. Okay. So fundamentally, what change control is, it's an evaluation of making a change before you make the change. It's about thinking about, is what I'm changing this to better? Is it going to have an impact? Is it going to make it better? Is it going to make it worse? And to me, that sounds a lot like a scientific hypothesis. You know, by doing this experiment, what's the outcome going to be? It's no different really with change management. So this is what I was thinking about. So when it's one person, one project, one protocol, then what I hear people say is, it doesn't matter if I make a change. Do you agree? That person suddenly leaves, they're ill, can't come into work. If it's all in their head and they know what they've tried, but it's not written down anywhere, then the next person comes in, and quite frankly, people do the obvious things first, and the obvious things to you will be the obvious things to the next person. So all that happens is they repeat everything that you've done and come up with the same conclusion. None of that works. Meanwhile, your PI is going, I need to get another grant because we've run out of money. Whereas if you just had a change control log, and all it needs to be, I say change control log, it's a spreadsheet with perhaps the protocol number as the file name, and then it lists all the things you've tried, when you tried them, who tried them, and you don't need to go into all the details, but perhaps whether it was successful, unsuccessful, it improved, it didn't improve, a snapshot, and where you would find the data to support that, whose lab book. Then you've got a history of your protocol development. Tell you something, if you go to Frederick's GMP lab with that, you'll be blown away, because he'll know not to try the things that didn't work and maybe focus on the things that did work when he comes to start to think about scale up. So it adds value later as well. Where it really becomes powerful is when you're progressing down this path. And okay, one project has been successful, you now take this further, and you're going to bring in more staff to work on it because it's becoming a bigger project. Now you've got a protocol, you're training different people on how to use it. Now person A makes a change to it, but doesn't tell person B and C. And person C makes a different change to it. And then suddenly you've got a process that's no longer reproducible because everyone's tweaking it slightly, but no one's talking to each other about how they're changing it. So in that way, change control can simply be, talk about this stuff in a lab meeting. I've changed this protocol for these reasons, and somebody else might go, that's a really bad idea because I've had this experience from before, or have you thought about this? Discuss it. That is you taking a change, deciding whether it should be implemented. If it's agreed, actually that's a good idea, let's change that. Document it in the meeting minutes of your lab meeting or a notebook. Lab book, have to be. And then you have done change control. Everyone knows, update the protocol, update your log, whichever system you choose to do, but everybody's on the same page. When you train the next person, then you're building in this kind of thinking about the impact of the change before you make it, rather than flying by the seat of your pants. When you get to GMP, it is locked in. It is more complicated to do change control, but that's for good reason, and that's because you don't want the protocols to change when you get to that point. But those initial change logs would have value for that process as well. Okay, I think I'm nearly there. I can't remember how long I said I'd talk to you, but my takeaways from today, please, are quality management isn't new. You can all do it. The principles are applicable from early innovation right through to product delivery. They just get a little bit more complicated, and your quality management system will start small and it will grow, but it will grow in a way that you can handle because you're familiar with it, you understand why it's growing, and therefore you're invested in it. When you try and impose a complicated system of somebody who's never thought about quality management, there's a lot more resistance to it. That's what I've just said. It doesn't have to be onerous to start with, but it massively benefits the initial investment. And there is an investment in quality management systems, not just the cost if you get a consultant in, or it does take longer. So you have to factor that into the working hours of your team. So you can't expect people to do everything that they did before and a quality management system on top, that you have to resource it. You get the best results when you empower and get the workforce to embrace it. So engage them in writing the protocols. Don't try and force it on them. Get them to develop it. My favourite story is I had a technician who used to work for me who was the most resistant person to coming on board with a GMP quality management system. She'd been a technician in a research environment, loved the fact that she could call her media batches Bart Simpson names, and was like, yeah. She now works for me as a consultant because she has tried to take the quality management system that I taught her and tried to implement it in different settings that she's worked in, because it gives her just some organisation around the different environments that she's been in. But she used to challenge me. So she would say, well, why are we doing it like that? I think there's a better way of doing that. And my response to that was, that's fine. If you can show me there is a more efficient way of doing this process for you, and it satisfies the needs from a quality perspective, then we can change it. Change control is not supposed to restrict improvement, it's supposed to enhance improvement. Leadership engagement is critical. And I'm sorry to say you will never finish your quality management system. It will always evolve, it will always improve, as it should. So once you start, so you will never stop. But that's not a bad thing. But I will stop. So these are my contact details, my X account handle, because it's not Twitter anymore, it's a bit old. Please feel free to reach out. I'm very happy to have a conversation now. You can challenge me on anything that I've said. I know it's daunting, but there are some simple things I think in there that you can take away.
Question, whether you had a view on, not specific, but how often SOPs should be reviewed, regardless of whether there's a critical incident that's covered by that SOP.
So we had a dynamic system for reviewing SOPs. And by that, I mean, there are some SOPs that once written, we reviewed biennially every 2 years. Things like how to use a piece of equipment, because that never changed really, unless you had a new piece of equipment or a different protocol on it. There are some things that change much more frequently because they're technical and improvements are found. So we had everything got reviewed annually, unless it was a piece of equipment or there was a certain number of changes associated with it. We did it that way. So we were only a team of 5 people. The thing that I did insist was reviewed on an annual basis at least was risk assessments. So every SOP should have an associated risk assessment, which should be reviewed to make sure the risks are still appropriate. But I think you can define that. In fact, thinking about it, I think we had a system initially where we released all of our SOPs as a working draft for 3 months, where you could test them. We weren't in a GMP environment then, so you could test to make sure that they were suitable. And then once they were locked into V1, version 1, then they were reviewed on their first anniversary. And then there was a decision made about whether it was every 2 years or every 1 year, depending. But that's defined in an SOP in its own right. Sorry, that's a really long way about saying you can choose.
Question, more of a general question again. From what you said about staffing costs, do you think labs have moved away from having kind of senior scientists who are involved in more of the kind of logistical things within the lab?
Mm-hmm.
Do you think we need to go back towards that would facilitate a lot of things like that?
Personally, yes. I think technical staff, the loss of core funded technical staff has been a massive blow. And a lot of this kind of stuff probably would have been supported by, I don't know whether you call it up north, they're technicians, they might be senior scientists down in London, but yeah, the sort of core facilities. Because if you're working with lots of little groups around you, you could have a system that everybody bought into centralised system. And then you've got more people to share the workload. I guess it depends how your institution would work, whether each lab would have its own quality management system seems a little bit onerous, especially if you have stores departments. You could, you know, ask store departments to implement some of these things around recording deliveries and lot numbers and checking C of A's, sharing that information or filing it so that everyone can access it, so not everyone has to have a copy of the C of A in a building.
Do you think, I mean, I don't know if you get a sense from other groups that you've worked with or funding bodies.
Are we going back to that? Is that what you're going
I
to
don't know what the answer is, but are they amenable to the inclusion of roles that would facilitate that, rather than just a postdoc that will work on this project?
I don't think they are amenable to it at all. I think it's very short-sighted of a university. I mean, universities weren't amenable to me being funded as somebody with all of this experience in building quality management systems. So I don't think well, I don't know. Core facilities should be, I believe, implementing these things because they have the services they provide have the ability to impact lots of people's research. So I think they should have a level of quality management in core facilities. I don't know if you have core facilities here or if you do everything yourself, but I think there is an argument that all core facilities should be having quality management training and implementing systems that show the traceability for whatever service they're providing.
I do not believe that is one of our courses that exists. We do have many, many courses, but I do not believe we have obviously we have risk management, an electronic system, but it's not at the level of SOPs, and we have the electronic data lab book system, but I don't think those two things are linked to quality of data recording. And we have archive systems, don't we?
But nothing's really standardized.
But nothing's standardized.
Group by group.
So I think it's
You're not unique.
gap.
You're not unique at all. And I'll be honest with you, when I was establishing RegenCTC, I spoke to 135 people about whether, as a business idea, it was needed. I think there was 2 people who said it wasn't out of 135 people. But the reason there is the gap in that market and there is the need is because actually there's no funding for it. It's a really hard place to be, because I think people think it will just happen at some point later. It'll be somebody else's responsibility. And it does get to the point where, you know, the investors have to go back and provide money, but investors are now starting to go, well, I'm not sure about this. They're getting a bit tighter on their due diligence. If you can't prove what you're saying with data, some of these things will help you with that as well. Yes.
Do you think a good place to target would be like the R&D office, where someone who has to register a project would then also be told part of this has to involve what your quality management system is going to look like? Because I don't think that's involved currently, and I don't know if that's people have to do that, but when we apply for R&D, it's always really challenging, but it's got nothing to do with like what sort of quality system we have.
So I am targeting technology transfer offices or research offices, because actually when you go to your tech transfer office or research office with a commercial idea, they do due diligence on it, but they do that due diligence from can we sell this in the marketplace? Yeah. They don't do the due diligence on where the research has come from, the document management system. And I think they should be. They should be helping people when they're writing translational awards, when they're writing MRC translational awards, DPFS GAP awards, DPFS awards. There should be something in there that allows you to start at that point definitely thinking about these things and putting these things in place. Before that, it's a nice to have. I'll be honest, you know, a lot of people won't have any of this in place. I personally think it's a mistake. I think if you have some of this knowledge in the background, you will improve your research. But I can totally understand when you're in a completely R&D setting, you're just trying to get the research done. Some of this stuff just feels like a waste of your time. And I get that. I've come across that. But I think once you're starting to talk to your TTO and you're talking about translational awards, then at that point we should be going, okay, how can we capture this development work to make sure that we're making the best use? Because all that happens is you get to the end of your DPFS, you go, right, Mr. Investor, I want this amazing widget. And they go, that's great, but we've got to repeat all that work because you didn't write any of it down. Such a waste of money. And not just a waste of money, but it's the time it takes to get back to the place where you were before. And that's why some of these development pipelines are 15 plus years. So they're just in the same place over and over again. So it's a personal opinion.
So I guess like quite often in some of these grant applications, there's a big section on data management, and a lot of the ones I've written and read, it's kind of like this will be saved in Excel format and GraphPad, Prism, whatever. Really, what we should be writing is all of that, right?
Yeah, I guess so. Yeah,
because that's how we'll manage the data to mean that it's good, solid data to then be taken further down the translational pipeline.
like I would like to see that. I can't say, if I'm totally honest, I don't know whether an MRC panel will read that and go, they understand what that means, because actually the funders I have a problem with. The number of GMP facilities I've seen them fund and set up, which has got no mention of quality management in them at all. They're just, this one random technician is going to put in an entire quality management system in 6 months. Like, they've got no awareness of what is needed at all, and they keep making the same mistake. Something that I have a constant conversation with them about. If anyone sits on panels reviewing those things, just think about that. Because you have the power, it's the people who review the grants that have the power to say what gets funded and what doesn't get funded. But yeah, if you've got 20 scientists and one quality person who's probably paid at a band 6 or whatever, you ain't getting a quality management system implemented in 3 years. Took me 3 years, but then I also didn't know what I was doing.
Why you've raised an interest, you talk about grant funders, but also mentioned about investors. And the process for an investor is just as random in terms of time and whatever. But I've seen many times when somebody is invested in something, or a group have invested in something, and then it doesn't work the same as it did supposedly in the lab. And part of the problem is that the process that was done in the lab is at times so random that actually when it comes to saying, well, actually, can we reproduce this? It doesn't work, or it doesn't work, excuse me, to the same effect. And that's probably why a lot of it, there's a graveyard full of technologies and medicines that never actually materialize into anything because of that problem. But also the timeline to actually get a product from coming out of the lab or that sort of institution to actually then being approved by one of the regulatory bodies is so long, because the quality of the data often is so, I was going to use the word poor, but that's probably not fair, but sketchy. So yeah, I agree with you. I think if this sort of system, these sorts of systems were implemented a lot earlier, then maybe it would knock the legs out of some research projects, because actually it wouldn't stand up to the scrutiny of a quality management system. But on the other side of it, it would make sure that actually ones that did actually had a much better chance of successfully translating.
Well, industry, it's fail fast, isn't it? So if you can fail something and get rid of it out of the developmental pipeline because you know it doesn't work, and you can do that quickly, you can concentrate on the ones that do.
Well, that's as well something that's changed significantly in the last 10 or 15 years, is that farmers now no longer do this massive blue sky thinking research and have pushed it out into universities.
Yep.
So they can fail, and then actually the farmers or the other companies will then mop up the good stuff. And you can see, without getting political, this government is wanting to change that. So actually, no, industry needs to take, or maybe the NIHR have shown their colours in a way by saying, you need to take more responsibility for this. And industry needs to get more involved with this. So I can see an even bigger disconnect coming between the two things.
It's quite interesting actually, because I think there's a disconnect between what industry think is done in academia and vice versa. So when I went to a conference workshop recently, there's a whole load of AstraZeneca ex-employees who are now doing different things in the room, and they were talking very generically about, yes, when you've developed this, we want all of your data, blah, 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 blah. And I was like, hang on a minute, what does all of your data mean? Well, we want all of your data. I'm like, yeah, but to an academic researcher, that means they want to know the results of your experiments. Yeah, and we want your traceability, and we want this. And it's like, well, that isn't what that means to a group of academics. So a group of academics will give you the results of the experiments and all of the analysis, but they won't have collected all of that other stuff because they don't know that that's what you need, because you just go, gives you data package. And there's just a disconnect between what that simple word data package means between those two groups of people.
I'll go through exactly that scenario at the moment. Exactly as you've described.
Yeah. And it's hard to find. I've been asked recently for, I can't remember who I was talking to, but a batch number of some fetal calf serum that was used in the lab in 2004. And they've asked me to prove whether that was used on this cell line or that cell line.
So can we just go through, just for the purpose of completeness, what is in the data package?
I don't know.
Because obviously, you know, I'm starting with it's the results and the data and the interpretation. So just go through it for everybody.
I think it kind of depends really on what your product is. And I was having this conversation earlier. So you can take it to an extreme, and as Frederic was saying earlier, when they're in the GMP lab, they have to prove everything about their facility. They have to prove that their piece of equipment works. So if you take that a little bit of a step back, you know, making sure you've got service history for your piece of equipment that is, and I would say you're critical. So you're not going to be able to do everything in the academic lab. You're not going to be able to capture every single piece of information that like they would in a GMP facility, and nor would you be expected to. But you should have looked at your processes and define which you think are your critical things. So which is your critical piece of processing equipment or your critical piece of QC equipment, for example, and make sure that you can prove that they are measuring whatever it is you're measuring accurately before you measure it for a result.
Okay.
Things like that.
Well, yeah, so I mean, we wouldn't naturally collect that, would we?
No.
So not just the assay and the data and the full SOP, but actually the equipment that it was run on and the service history of the equipment that it was run on. Is that what that would be?
That's useful, yeah.
That would be part of the data.
Yeah.
And the people who did it, or
People who did it, yeah. Whether they were trained to do it, what their training
records,
the records of those things, yeah. Now, and they could be
further.
well, so things like, you know, the reagents that you use on it. If there are critical reagents,
that's fine. I think
yeah,
think of that. I don't know.
That.
Yeah. Okay.
So I have I worked at a company as a postdoc.
Yep.
I was allowed to not record anything as the company would. I would never finish my postdoc with what was my aim, because it takes a lot of time. You agree? And you say that it is a point. And there they actually, for the company, they have to then go for patents and everything. So just to answer to Jane, for example, every time they were preparing, there were a company working for cell therapies for Parkinson, Genetic Biotech, that's the company. And for every time they prepare a media, then they had to say the catalog number, the batch of each product that they used. They had tables where they recorded each single data, every piece of it. I used this machine, catalog number, and this was all recorded every single day for every single thing they did. It's a lot of information that if we want to go for this, then the scientist needs to put a time to collect. And then each page of a lab book was contrasigned by a colleague. So the technicians, they were taking time every week to take pages of a lab book from another colleague and stamp and sign that they've been checked for clarity. Is this what?
So this is what we had to do when we were working on our license towards licensing, yes. And I think you can pare it down to start to introduce it early, but with a view that, I guess what I'm trying to say is, if you start there, it's a huge mountain to climb. And what I'm trying to encourage people to do is start thinking about some of the things earlier, because once you start, actually you kind of get a bit of a bug for it. And people who hated quality management suddenly develop a love for the organisation that it brings. And the difficult technician that I talked about earlier, she got on board when she was able to discover that just by recording the temperature of the -150 freezer on a monthly basis, just a monthly basis, she was able to detect the fact that the compressor was beginning to fail, and we got the engineer out and it was repaired before it failed. Now, that's huge when everything you have is stored in that one freezer. So, you know, there are, if you collect data, but you review, and this is what I mean by having to retrieve the data and trend it and look at it, that has to be somebody's responsibility to do that, because otherwise it's pointless writing that number down every week if no one looks at it.
But you need a data analyst to do this kind of job.
You don't necessarily need a data analyst, you just need somebody who can look at something and go, no, it's getting warmer, or plot it on a graph. You know, it's like, you know, you can take it as far as you want to take it, but I think at the moment, no one does anything. And that's what I'm trying to change. Because I think some of these things can easily be incorporated if we just, and again, my same technician, she said to me just last week, I wish somebody had told me then that what I was writing down was important now, because I would have recorded it, I just didn't know I needed to. So you think about your experiments in the now. So they used to record, they used to make their media and give it all stupid names so that they knew that Bart Simpson had been given to researcher A and Maggie had been given to researcher B. But for them, that was about tracing contamination in the short term. Who was contaminating their media? Which was a very sensible system at the time, until I said to her, yeah, but what did you put in each one of those bottles? She's like, I don't know. So I know Bart Simpson went to Neil, but I don't know what Bart Simpson was made of. So yeah.
I think, yeah, I can attribute, I moved from an academic lab into a clinical diagnostic lab, and it was the biggest jump for me ever, going from no quality management system to a full on every single thing recorded, as you were saying. And initially, I mean, it's taken me over 2 years to fully be on board with everything that they do, because I initially thought, well, I'm doing research, why do I need to do all of these things? And now I really appreciate so much more why everything is really helpful. And I used to make silly mistakes with just like numbers and things, and then be like, oh, I'll just repeat that, that's fine. Now I'll have a worksheet where I check and someone else checks, and then I don't make that mistake anymore. And that saves time, money, everything. So I think, yeah, there's a lot to learn. But if anyone doesn't know what you can implement, just spend a day or two in a clinical diagnostic lab and you'll find out exactly what processes are implemented. But then obviously we're lucky because that's been implemented at a higher level that everyone just follows, and there's someone in charge of doing that, so it's easy. And I'm sure it took a long time to get to this point, but there are lots of things that you can do that are small to start with, definitely.
I think one of the traps that people fall into is saying, I need a quality management system, and then they Google it. Yeah. And you can download loads of templates for quality management. But what makes a good quality management system is that it does what you're doing in your environment, and it captures the information. So you have to understand why you're doing it. Because if you're filling forms in for filling forms in sake, nobody sees the value in that. And the reason that most people have findings on their ISO inspections or their licensing or audit inspections is people not following processes, because the processes actually are not what they do. What's the point in that? Just have the process that you actually do written down, so get the people who do them to write them down. So anyway, I'm sticking around, so I'm very happy to have chats with people. Yeah.
Thank you so much, everyone, for coming. Thank you, Zoe. If everyone who's here has filled out their email, then we can share the slides and record. Well, the recording will be uploaded to the BRC website. So, but yeah, I think we have tea and coffee now. Please, yes, please do the survey as well. And Zoe also wanted to create, do you want to?
Yeah, I might send it to yourself if that's okay.
Okay, yeah,
What I'd be keen to hear is how you felt my delivery of the seminar came across, whether it was pitched at the right level, whether you found it useful, whether you think there was things that were missed that you would have liked to have known about. That would really help me out and the next group of people that I do the awareness training for. So thank you.
On your way out, on your way out, please can you scan this and do it so we have some more information for our own EDI as well. Yeah, otherwise let's go upstairs again and hopefully have some tea and coffee. That's not too cool. Thank you.